Bismillahirrahmanirrahim. We will discuss endocrine system today. This system has to produce some secretions. We name them as hormones. And what it has to do, just like your nervous system, the endocrine system also coordinates your body growth and development, right? And what they do, they take information from your peripheral cells and organs. And in the nervous system, we can see that the nerve impulse should be a reason to proper function, right? And what happens in your endocrine system, that its hormones have to reach some target organ, right? And for this, they have to reach through various tissue spaces and your vascular system, right? And we should know that these functionally interrelated, right? And your endocrine system response that it produces is slower and much prolonged, right? Rather than your nervous system that is very quicker in its response, right? But what happens that they simultaneously have to approach the target cells, all the tissues of your body, and in this way they function properly, right? In this picture, as we can see that there are three modes through which your endocrine system has to function, right? What these modes are. The hormone, which we know as a biological substance, it definitely has to reach your target cells, right? And when it is being secreted, in the first picture, you can see that it has to reach towards your circulatory system. This is its one way through which it has to reach the target organ. In the next picture, which shows the paracrine or the juxtacrine communication way, in which what happens that the hormones, they have to diffuse in the extracellular fluid to reach the nearby, you would say, or we can say some distance away located target cells to be acted upon. In juxtacrine, if we mention, they have to act upon the cells which are just adjacent to them, right? And in the C picture, you can visualize the autocrine mode through which the hormones act. What happens here? The chemical messengers that are being produced by the cells, they should bind to the receptors located on the surface of the same cells. So we should know the mode through which these hormones are acting, right? Now look into the picture. The chart is showing you that there are various varieties of the hormones that are being secreted. First of all, see the amino acid hormone derivatives, right? You can see here predominantly the thyroxine, the adrenaline, and you can add the melatonin, right? And then you can see the list of the steroid hormones that are being derived or produced by your gonads and adrenal cortex, right? You can mention cortisol, and you can mention aldosterone, androgens, progesterone, testosterone, etc., right? And if we talk about the polypeptide hormones and small peptides and protein, you would say I can mention the ADH, the oxytocin, insulin, pituitary tropic hormones could be there, calcitonin, then you can mention the prolactin, parathyroid hormone, growth hormone, and lipoproteins. FSH, you can add ACTH, I've mentioned, right? And next to it, we have to see various receptors, right? Now these hormones have to reach some receptors they have to find, and they have to act on the target cells by binding to the specific hormone receptors. In the picture, you can see the cell surface receptor, and here the hormone, which is protein in type, they are supposed to attach on the cell surface receptors, right? Because they are not supposed to pass through the cell membrane. And here you can see that they need the second messenger, then they produce their physiological effect. Now concentrate the other variety of the receptors. These are intracellular, right? And in this picture, you can visualize that fat soluble hormones like steroids, they easily pass through the plasma membrane of the target cell, and after binding the intracellular receptors, they produce the effect, right? And your endocrine system consists of cells, tissues, and various organs forming the glands, right? And they have to synthesize and secrete the hormones. in the list of glands, we should first visualize the master gland called as Pituitary gland, and another type is pineal gland. And we can see in the neck region the thyroid gland and the parathyroid gland. And on the superior pole of the kidney, the adrenal gland rests. And if we want to see the collection of various cells and tissues, we can visualize the pancreas, where islets of Langerhans function to produce hormones. And we can see that your reproductive organs, the ovaries and testes, are also supposed to produce the hormones. And the another type of variety, which you call it as the diffused neuroendocrine system, that includes various endocrine cells that are located in the mucosa of your digestive and respiratory tracts, right? And they are scattered among other exocrinal secretory cells to function as endocrinal cells, right? Now we have to talk about the pituitary gland, which is a master endocrine gland because it has to produce various hormones that influence the activity of the rest of the endocrine glands, right? Here you can see a pea-sized yellow color pituitary gland, and it is being suspended from the hypothalamus by a short stalk, and you called it as infundibulum, right? And we should know that it has got two main subdivisions. For this, we have to discuss little bit of its embryology, right? We should know that your pituitary gland that has got the anterior lobe called as adenohypophysis and the posterior lobe called as neurohypophysis. These both subdivisions are derived from two different embryological sources. So we have to discuss them first. And here in this picture, you can see that there lies the neuroectoderm, and your neurohypophysis is derived from it. What happens that first of all, the neuroectoderm arises as a ventral evagination, right? And from where? From the floor of your diencephalon, which is a part of your forebrain, right? And from below, you can see that your the oral ectoderm or oropharyngeal ectoderm is going to form your outpocketing from the oral cavity called as stomodium of the embryo. And this outpocketing is also called as Rathke's pouch, and it grows superiorly or cranially and approaches to the neurohypophysis, and it has to make connection with the forebrain by you would say means of the infundibulum, right? And in this way they are supposed to form the complete the gland. And here in this picture you can see the approximation of the neuroectoderm and this ectoderm of the oropharynx, right? And where they lie, they basically occupying the bony part of your skull, the sella turcica, right? Now here you can see the developing sphenoidal bone holding the pituitary gland, its both lobes, the anterior lobe and the posterior lobe, right? Again we can see another picture that your pituitary is lying in the pituitary fossa or the sella turcica, you would say, and it has got the anterior and posterior lobes. The posterior lobe you can see is hanging by the infundibulum and is attached superiorly to the through the median eminence to your hypothalamus, right? And this is the anterior pituitary, and we will discuss its three parts. It's being divided into the most prominent distal lower part called as pars distalis, right? And then you can visualize its tubular superior extension that is winding round the infundibulum as a collar. This is called as pars tuberalis, right? And then you can visualize the pars intermedia, which is present just posterior to the pars distalis and anterior to the posterior lobe, right? Now visualize the posterior pituitary or posterior lobe that has got the extended lower part called as pars nervosa and the upper tubular stalk-like structure called as infundibulum, right? Now you can see the details of anterior posterior pituitary. Focus on the anterior pituitary. the three parts, the pars distalis, the lower one, and its upper extension, the pars tuberalis. And we see the bluish color part, the pars intermedia, which is lying behind the hypophyseal pouch or the remnant cleft, right? So for this, we have to concentrate that the anterior lobe or adenohypophysis is divided by the residual lumen of the Rathke's pouch into two unequal parts, right? The pars distalis almost make 75% of the gland, and it lies anterior to this whitish cleft, right? And the posterior wall of this Rathke's pouch, it extends and lies behind the cleft, and it forms the pars intermedia. So it is showing thin cellular partition between the pars distalis and you would say the neurohypophysis behind it in light brown color, right? And what else? You can see the pars tuberalis that extends upwards towards the infundibulum and hold it like a collar, right? That is the detail of the anterior lobe, right? Another good picture to show the embryological growth of the pituitary gland. In the week 3, you would be observing the hypophyseal pouch, and here you find the stomodium, which you call it as future mouth, and on its superior aspect you can find, let me show you, the neuroectoderm that is forming the neurohypophysis, right? And in the below picture you see that how they both are approaching towards each other, right? And in this way they are also having a cleft like fissure that should remain in the middle portion of anterior pituitary, right? Let me make it clear from this very picture. This is the third picture showing you the neurohypophysis and the adenohypophysis, right? And finally you can see that both these are approaching towards each other and resting in that sella turcica to form the pituitary gland, right? Now from this picture again we can see that there lies the hypothalamus superiorly, and we can visualize the infundibulum, and we also see the anterior pituitary and the posterior pituitary. The anterior pituitary is going to form various hormones, which I can mention you that there are 5 types of cells in your anterior pituitary. They are supposed to produce hormones, and their names are given according to their hormonic production. So first of all, there lies the somatotropes. These almost make the 50% of the cells, and what they have to produce? They have to produce the growth hormones, or you call it as somatomedins, right? Or another word is being used, the somatotrophins. They are the hormones produced by your anterior pituitary. There is another variety which you call it as mammotropes, and they are again produced by the anterior pituitary, and they act upon the mammary gland, and the hormone being produced by these mammotropes or lectotropes is the prolactin, right? And they constitute 15 to 25% of the cells of anterior pituitary, right? Now we have to concentrate on the corticotropes that are again 15 to 20%. What they do? They have to produce ACTH, right? And in this way, you must know that they are acting upon the adrenal cortex, right? And then you reach towards the gonadotropes, which includes the hormones they are supposed to secrete are FSH and LH, and they are gonna affect the female and male reproductive system, right? And then we should visualize thyrotropes, which make 5% of the you would say cell population of anterior pituitary. They have to produce the thyroid stimulating hormone, and they have to act upon the thyroid gland, and so they are also called as thyrotropin. In this way the somatotropes, the mammotropes, the corticotropes, the gonadotropes, and thyrotropes. These are 5 types of cells being produced by your anterior pituitary, right? Look into the cut section of this pituitary gland. This is the pars distalis, and the cells we just discussed right now. And here you can see the cleft, and on its posterior aspect you can find the pars intermedia, that is rudimentary and basically consisting of some follicular cells that are lined by the cuboidal cells, and the colloidal material is there, and few cords of basophilic polygonal cells be a part of it, right? And they are supposed to produce the hormone called the melanocyte stimulating hormone, right? And in this way they stimulate the melanocytes to secrete the melanin pigment that are lying in the epidermis, right? And now if you concentrate, that is the pars tuberalis, and it forms a collar-like structure around the infundibulum, and it's being filled up of you could say groups or cords of cells that are showing faintly basophilic staining. Cells are cuboidal, and you can see so many blood vessels present here, right? And here the cells are supposed to produce LH and FSH, right? And here you can see the posterior pituitary or posterior lobe, which is showing in its histological view that it is made up of unmyelinated nerve fibers, right? You call them as axon, and there could be the fenestrated capillaries along with them. And you can find specialized type of glial cells which are called here pituitary, right? And these axons we find here are about, you would say, millions of them, and they originate mainly from the neurons which are located in the hypothalamus, and we name them as supraoptic and paraventricle nuclei, right? And their axons basically are converging at the median eminence, and there they form the nerve bundles to form the hypothalamo-hypophyseal tract, and this has to pass through infundibulum, reaching to the pars nervosa or posterior pituitary, and here at their dilated terminal end they are in close proximity to that of the capillaries. So the dilated terminal ends of the axons, called as, we name them the herring bodies, and they are basophilic masses, and all of them are storing the hormones we label as oxytocin and vasopressin, which is also called as ADH, anti-diuretic hormones. And we should know that this pars nervosa is just storing these hormones, because these are not being produced by the posterior pituitary, rather they are produced by the hypothalamus, where the nuclei supraoptic and paraventricle, they are going to produce these hormones. They are just sending them down through the infundibulum and then reaching the pars nervosa, right? And if we can concentrate, there lies the glial cells that support the axons here, and these are small polygonal cells, and could be showing various cytoplasmic processes, and they are wrapping around the axonal fibers, you would say, or their terminal dilated. Now you can see the schematic representation of the pituitary gland, and starting the introduction of this pituitary gland, we should focus the 4 C's. The first C is the connective tissue capsule, which is present all around, made up of dense irregular connective tissue. Then we can visualize the second connective tissue septa that are entering inside to divide the inside parenchyma into various lobular pattern, right? And the third C is depicting various cells that could be in the form of squad, clusters, clumps, and so on. And the fourth C is showing the fenestrated sinusoidal capillaries, right? So 4Cs must be a part of any endocrine gland that you should mention. First of all, the capsule should be there. Second is the connective tissue septa should be there. Third is the pattern of cells that could be in a form of always clusters, clumps, columns, or anastomosing cords. And 4C is showing the capillaries as fenestrated sinusoidal in variety, right? And if you concentrate the whitish cleft, and behind it you can see the pars intermedia, and that is showing various cells in a form of a cords, and they are polygonal cells, right? Now look into the histological cut section of the anterior lobe, and here we are supposed to look into the two varieties of the cell type present here. One variety is chromophobes that are making 50% of them, and they stain poorly because their cytoplasm is not showing granules that could have taken dye, right? And they are behaving as a resting secretory cells, right? And the other varieties are chromophils, and we divide them into acidophils and basophil. Among them, acidophils have strong affinity for acidic dye, so takes eosinic stain under light microscopy, okay? And the basophil cells, they have got cytoplasmic granules, and affinity for basic dye is very much high, so they show the bluish tinge or hematoxylin stain when we observe them under the light microscope after H&E stain, right? And these acidophil basophils, that forms the chromophils, again forming 50% of them. And among them, acidophils are 40% and basophils are 10%, right? Let me show you various chromophils in anterior pituitary and see various letters. The letter S are the somatotropes that make the 50% of them, right? And these cells could be oval in shape, having centrally placed spherical nucleus, and their cytoplasmic organelles are full of secretory granules and take deep acidophilic stain, right? And the hormone, as I have to mention, they produce is the growth hormone called as somatotropin, right? Then see the letter L that is placed below, making the lactotropes or mammotropes. They make 15 to 20% of them. These cells are polygonal in shape with oval nuclei, right? And their cell organelles are also filled with the numerous large acidophilic granules. And they are supposed to produce prolactin to act upon mammary gland and the uterus, right? Then you have to see the letter C showing the corticotropes. They make again 15 to 20%. These cells are polygonal, having rounded nucleus but located in its eccentric position. Right? See the letter T that is marked on the thyrotropes. They make 5% of the parenchyma. They are large polygonal cells with eccentric nuclei. They are supposed to produce thyrotropic hormones, means thyroid stimulating hormones, right? They take basophilic stain. So among these 5 cells, let me revise that somatotropes, lactotropes, they both are giving acidophilic stain, right? And the corticotropes, gonadotropes, and thyrotropes, they are the basophilic staining cells. We can see that the pituitary, which are glial cells, their processes are not taking good stain. Rather, their pale staining nuclei can be visible under the light microscope, right, in H&E stain. And after that, we have to concentrate on another picture. Here in this picture, you can visualize the paraventricular and supraoptic nuclei of the hypothalamus. They are supposed to produce oxytocin and ADH respectively. And you can see that from these nerve cell bodies, how axons are descending downward, passing through the hypothalamus, and the infundibulum is their way to travel to reach the pars nervosa, right? And after that, you have to focus on the blood supply of the pituitary gland. That is very much important because all the hormones which are being produced are supposed to enter into your bloodstream. So concentrate that there is the superior hypophyseal artery, which is a branch of internal carotid artery that is winding around the infundibulum or median eminence, and here they are supposed to form the primary plexus of the hypothalamo-hypophyseal portal system. It means two capillary plexuses exist. Number one is this primary plexus, and they are gonna form the portal hypophyseal veins, right? You can see them in blue color, and they later on form the secondary plexus of that hypothalamo-hypophyseal portal system, right? And here you can see the connection that they are gonna drain by the hypophyseal veins, and also you can visualize the hypophyseal veins on the posterior pituitary, and plexus lies here also, and from below the inferior hypophyseal artery is gonna join it. So there lies the two plexus, the primary and the secondary capillary plexus, and there lies the, you would say, hypophyseal portal vein in between the two, and then you can observe the hypophyseal vein also at the lower end. So this is the venous type of portal system that exists in your pituitary to carry the hormones towards your body to other target organs, right? Now in this picture, histological view, you can visualize the chromophobes that are not getting stained well due to less affinity for a dye, because they don't have that much secretory granules rather, right? 50% of them. And then you can visualize the chromophils, which are acidophils 40% and basophil the 10%. You can see various granules and their staining color, right? And pituocytes are also visible in the last picture, right? We have got a very beautiful picture to learn the negative feedback mechanism, and for this we have to learn the relationship of hypothalamus, interior pituitary, and the thyroid gland. First of all, say if the body is cold or having the low temperature, that is being sensed by your hypothalamus, and it is either stimulated or inhibited. Here it gets stimulated to release the thyroid releasing hormone that is acting upon the interior pituitary, and it will be producing thyroid stimulating hormone, and then it in turn act upon the thyroid gland to produce the thyroid hormone. Then this thyroid hormone that stimulates the target organ activity and increases your body temperature. And finally, when your body temperature is increased, again sensed by your hypothalamus, and the thyroid releasing hormone is no more there, and finally the thyroid stimulating hormone will not work, and finally thyroid secretion will be decreased, and this negative feedback mechanism exists in your body to control the temperature, right? This is the slide you will observe in your lab. There is a whitish cleft you can see, and anterior to that cleft is the pars distalis, right? And posterior to the cleft, this is the pars intermedia, and just behind it you can see the different histological features. Yeah, you are in the pars nervosa. In this way we can learn the slide easily, right? There is another schematic representation of various cells. The unstained cells are chromophobes, the pinkish stained cells are chromophils, and basophilic stains are basophil cells, right? And here you can see the pars nervosa, and the letter P is showing the pituocytes, and the word NB are showing the nerve terminal dilatations, right? And which are being stained, and you can call them as herring bodies, right? Again in this slide you can see the basophils and the acidophils and the cutted blood vessels that are running into the septa in between the parenchyma, and this septa, which is connective tissue made up of reticular fiber here and having the fibroblasts in them. And what else you can find? You can see the unstained cells called as chromophobes, and you can see the lumen of the capillaries, and the bluish tinge is taken by the connective tissue septal fibers, right? Another histological slide showing you the basophils, the acidophils, and the chromophobes, right? It's all for today.